Good evening, everybody. Today we are going to start the next chapter, that is bioinorganic chemistry. So, bioinorganic chemistry actually it is a science in which biological phenomena are analyzed in terms of chemical principles. We know that all the biological molecules are mostly organic compounds, and we can study there is a recent studies on biological molecule and biological activities and energy required for the life process. So the biochemistry actually it is the combination of study of bioinorganic molecules and analysis diagnosis of this different activities of the biological activities taking place in our body, also in living organism, and analysis of the certain biological parameters, etc. Now we know that there is a wide range of metal and non-metal is present in biological systems. So the life actually depend upon the inorganic chemistry and organic chemistry, physical chemistry, and analytical chemistry. So these are the different combinations or different branches of the chemistry, and if you are we can use these branches for study the biological molecules in our life. So the essential elements means there are several elements are essential for maintenance of the life in organism. These elements are called essential elements. So the essential elements can be divided into two different types. One is bulk element, the second one is trace element. So bulk element can be again divided into macronutrients and micronutrients. Macronutrient means the element which we have to need for a large quantity. For example, oxygen, carbon, hydrogen, nitrogen, sulfur, phosphorus, etc. Then micronutrients means we need little bit lesser amount of the substance, lesser amount. For example, iron, copper, zinc, manganese, molybdenum, etc. And the second variety is the trace element. Trace, ultra trace elements means we need only little amount of the element for the biological activity. Now we are moving into the individual term bulk and trace metal ions. And these metal ions act as a cofactor in enzyme in our body, or act as a structural entity, or used in controlling of metabolic pathways and other mechanisms, or as an oxygen carrier, or it is used in the maintenance of osmotic pressure, pH, and regulatory action in our body. Now we are moving into another term, that is conjugated protein enzyme. So a conjugated protein enzyme consisting of the two parts. One is a non-protein portion and it is known as cofactor, and the second one a protein part and it is known as apoenzyme. See here here I have shown a figure. So in which the orange one is the apoenzyme, that is it is a protein part. If you are clubbing with a cofactor, that is non-protein part, we will get a haloenzyme. Okay. Now a cofactor is independent and it is independent for catalytic activity of conjugated enzyme. Say for example Mg2+, Ca2+, Zn, etc. But the enzyme which is tightly bounded with metal ion cofactor is called metalloenzyme, and the metal ion in the enzyme is contribute towards the structure or catalytic membrane or both. So one example is alcohol dehydrogenase, and it consisting of the 2Zn2+, and it will contribute towards the structure and catalytic metabolism. Thank you.